So thank you, Dan. We give you the stage for your talk. Thanks.
Thank you.
Modeling talk.
Thanks for the opportunity. So as Thomas gave us a nice introduction, me and Ali, we're doctoral students at the University of British Columbia, and we started collaborating and then started through discussions to talk about sociality in microbes. And sociality in microbes, it's an interesting kind of topic because as you will see, not only it's harder to imagine than sociality in animals or humans, but it's hard actually not to use the concepts and the words that we already use for animals and humans to talk about microbes, even though the fit is a little bit strange. So basically the whole what we are applying our thinking to in the real world, as it were, is this phenomenon of ocean debris being degraded by bacteria. So this is something called marine snow, and I included here a picture in the first slide. This is a piece of ocean debris. It could be something like a dead plankton or a fish fecal pellet, or some dead pieces of stuff that's floating in the ocean, and bacteria adhere to it, and they release sticky polymers that causes different pieces of debris to stick together and get bigger and bigger until it reaches some critical mass, and then it starts sinking. So the reason it's called marine snow is that you take a flash photo inside the water, it looks like it's constantly snowing. There's all this debris just sinking. And this phenomenon is there's a lot of what appears to be cooperativity going on between the degrading bacteria. And this cooperativity is in several different aspects. So one of the important aspects is this rate of the degradation. The bacteria are releasing these enzymes. Basically, bacteria can't absorb the food. They have to release their digestive enzymes outside. They don't have a stomach. So they release the extracellular enzymes outside, and they cause a release of nutrients from this debris, which is a complex aggregate of material. And the liberating all these nutrients causes this huge excess, or what I'm trying to say is they are degrading the marine snow particle at a rate which is far greater than the rate that the bacteria are actually consuming on the particle itself. So it leads to generating this large excess. And as the particle sinks, a lot of our understanding of this phenomenon has come from detailed physical simulations and photographs of scuba divers. But basically, there's a picture of results of a simulation that shows this excess is basically trailing as this plume of cloud of excess nutrients as this particle is sinking in the ocean. There's all this excess nutrient. So this, to an evolutionary biologist, seems like a cooperative phenomenon. So they're degrading it faster and much more than their individual needs. And then there's another aspect of this cooperation in which all of these, as I mentioned, the particles are very complex, and they contain various different types of polymers depending on what the origin of the waste is. So there's many types of nutrients that you can liberate from it. And but the bacteria can, in fact, help each other or feed each other different types of nutrients and have this kind of complementarity. So here in this cartoon, I drew these ellipses are bacteria supposed to represent. And then there are these two different nutrients, this yellow and brown nutrient. And these two gray organisms in the middle, they don't produce both the yellow and the brown. Instead, they cross-feed with each other. So these type of bacteria are called, in the literature, they're called syntroph, or synergistic trophy, synergistic eating. That's where the word originates. And in some cases, we do know that you don't have to share. There is not some reason that you cannot produce both goods. So there are these self-sufficient organisms or prototrophs that also exist. And then there are organisms that don't produce any of the goods, but if they are around, because these goods are just freely diffusing, these nutrients are just freely diffusing out of the bacteria, they can take in all of these produced goods from the syntrophs without paying any of the cost of the work necessary to produce these goods. So we know that the self-sufficient organisms exist. We know that free riders exist and potentially can arise by mutation. So here is this where the sociality aspect of it comes in. We have these words like cooperative and free riding. So we have something that appears for the public and like altruistic or cooperative, and some organisms that appear selfish. And these words are taken from, as I mentioned, theories that originated from people thinking about animals and humans. So there are very loaded words like free riding, and there is an interesting way in which this we think that thinking about bacteria makes it really clear what the evolutionary theory is trying to do, which is actually not to label these strategies as such, in particularly in this case. And in order to explain a bit more what I mean exactly by that,
then
yeah,
little, just go back 1 second, just to clarify.
Yes.
I'm trying to make sure that it's clear that this guy here is self-sufficient. This guy doesn't need anybody to survive. These guys need each other.
That's right.
Just
yes.
one produces the what, so they sort of, we will say, they cooperate, and this one just profits from the work of others. So
yes.
These are all the landscape of possibilities. Okay. Yeah.
Yes.
Yeah.
And if we want to talk about it from the point of view of dependence, then actually this has to be a complete system. Like the free rider can't exist without, or it could be a free rider and a prototroph, because the free rider has to get the goods from somewhere, right? So the system, because free riders always can arise by mutation, and they always can arise by migration, we can't really rule them out. Then whenever you consider a system like these two, you have to consider in a context, and the context includes organisms that don't participate. Does that make sense?
Yeah, thank you, because that's what gives the sociality aspect clear. Yeah, thanks.
Yeah, okay, thank you. So then in evolutionary terms, we're looking at the frequencies of the different types. And then ideas like stability and fitness all arise from looking at how these frequencies change over time. This free riding as a term, or selfishness as a term in bacteria that don't cooperate, I have an interesting kind of anecdote or historical example of that will kind of maybe help to frame the problem in the way that we would like to people to see it. And this historical example originates actually with a completely different model. It's a very famous model that you may have or not have heard of. It's by a German mathematician. His name was Breas, and the Breas paradox is the name of this phenomenon. It's basically you have drivers that drive on this network of roads, and they try and get from A to Z here. And the paradox basically says that under some kind of under a wide range of conditions, particularly if some of the roads are kind of like highways that doesn't matter how many people drive them, they don't get congested, but some of the roads aren't like this, so their congestedness depends on how many drivers there are. You get to this scenario where if you have just the diamond without this road between B and C, if you just have these two roads to get from A to Z, you're actually better off than if you added this extra road. Or in other words, if you remove an extra road, you can actually relieve congestion. So that's where the paradox comes in. And it's kind of like this framed in this game theoretic sense that every driver chooses the best road for themselves. So this kind of this that it's written in the article, and it's written kind of in the way as the code of the way to think about this problem is that you have to assume that all the drivers are maximizing their self-interest, and that's what leads to this paradox, because in the end everybody choosing the best thing for themselves leads to a worse outcome overall for the group. And what's very interesting is that many years later, people have come up with analogs of this exact problem that don't involve any drivers or people at all. So Cohen and Horowitz wrote an article where they showed that if you have a system of strings and springs, you can achieve a perfect analog of this paradox. Basically, you have here you have a string and a spring, and then here you have the opposite arrangement. So it starts off with a spring and then a string, and they both of these ropes, combined composite rope things, balance, like they hold a mass from the ceiling. And then if you cut off this rope, then what actually happens is that if you cut off this rope, the mass ends up hanging higher than it originally did. So it's kind of an analog of this traffic paradox, that it says that an extra, what looks like an extra support, so an extra wire, actually makes the whole system worse at carrying the load. I hope this is clear, the analog between these two. If not, please let me know, but maybe you've heard of it. And I think the interesting point about this kind of translation of this problem of traffic into a mechanical analog is that, of course, in this case, there is no analog for selfishness. You don't have to have any assumptions about selfishness and that the individual units of whatever the tension are perfectly selfish. So there's something kind of unavoidable here, and it says something about the role of the drivers, and the role the drivers play in this particular model is that the drivers, if they have a road that has less resistance than another road, then their consequence of choosing that road is kind of it's like a least resistance thing rather than a choiceful strategy. So it's almost like a thermodynamic consequence. And then when you add and remove roads like you do in this paradox, you're basically doing something top down. So you're changing the options that are available. You're changing the structure, and the structure is what results in these thermodynamic particles causing the structure is what results in the overall outcome. It's not really the fact that the organisms or the drivers or the tension is choosing one way or the other. So this top-down effect is the effect of structure. And in bacteria, you can also say if a bacterium produces a particular good and another bacterium is next to it, if a bacterium has access to food, he will just eat the food. It's not really something that you can consider a choiceful strategy that is implied by words like cheating or cooperation. So, or in other words, for evolutionary theorists, maybe I'm going about this because I also know in complex science, there's been people that have kind of reacted to the evolutionary theory presenting the cooperation so strongly in terms of these words like cheating. In this particular case with bacteria, what Ali and I want to say is that when you present cooperation in this framework in terms of cheating, all it does is that it makes the idea of structure or top-down structure of the environment an interesting phenomenon. It's basically asking you to define your structure. What's the structure that's going to solve the problem? And structure is just another word for saying if the bacteria, if I as a cooperator, how often do I get to interact with another cooperator versus with a cheater or a free rider? And so in a world where there's no structure at all, it's equivalent to saying that my frequency of interacting with free riders versus other cooperators or syntrophs is basically like Brownian motion. There's no structure at all. We're all just equally likely to hit each other and bounce off each other. So in biology, this is called panmixia, or completely free mixing. So the only time where free riding actually poses a problem is when the world is a complete one test tube that's being constantly shaken. But obviously this is not the case. And then the question of structure becomes interesting. And I provided here on the left an interesting paper that you may have heard. It's actually these are two professors at the University of British Columbia, where we are. And they created a spatial version of very famous games in game theory. One is called the Prisoner's Dilemma, it's on the left, and the other one is called the Snowdrift or the Hawk Dove game. So these are like cellular automata type of ways to represent the game. So they actually noted that okay, well, first, maybe I will introduce what these two games are. The main difference between the Prisoner's Dilemma and the Snowdrift game is that in the conditions when there is no structure at all, in the conditions of this panmixia, where everything is just Brownian motion, then there is no stable equilibrium of cooperation and cheating in the Prisoner's Dilemma, but there is an equilibrium between cooperation and cheating in the Snowdrift game. So that's probably the main difference in them. And maybe if you know about the payoff matrices, the way to get from a Snowdrift to a Prisoner's Dilemma is just changing one of the payoffs. What they noted was kind of just an interesting, so I just wanted to provide this example as a little bit of more nuanced understanding of how structure can affect social interactions, is that this Prisoner's Dilemma game, which is unstable in a free mixed environment, so you can either have only cooperation or only cheating, suddenly when you add space, cooperation becomes highly stable. And then these black clusters are clusters of cooperators. So cooperators, because they benefit each other so much, they like to stick together as big clusters. And that's how they become stable in a spatial game, or in a game where there is an ability to be structured, where you can interact with your neighbors more often than with other organisms or other cells that are further away. However, in the Snowdrift game, which, as I mentioned, the main difference is that the Snowdrift game is already stable. So you can have an equilibrium of cooperation and cheating in a completely unstructured setting. As soon as you add structure, it kind of starts to actually harm, in a way, the integrity of this interaction between cooperators and cheaters. Basically, what's happening in the Snowdrift is that the equilibrium arises from the fact that whatever the common type is doing, you would like to do the opposite. So there's always a benefit to doing what's rare. So in this case, when the game becomes a spatial game, when it becomes a structured game, then whatever your neighbor is doing, you would like to do the opposite. And so that breaks up these clusters, and it leads to this very fizzled out pattern, which actually causes the game to be less stable for a wide range of conditions. So I just wanted to provide a little bit of a nuanced example here. And then on the right, I just taking from a summary of just all the different ways in which bacteria can be structured in the environment. There's really so many different ways. Of course, they can be limited by space, so you only reach those who are very close to you, but there's bacteria can stick together and exchange information. They can recognize each other. They can grow in certain ways. So really the types of structures is the interesting thing, is what evolution is trying to ask you to clarify when it introduces this idea of free riding. It's asking you to clarify what's the structure in your system. So now, to frame our problem a little bit more precisely, we were talking about the marine snow being cooperative in the sense that it leads to this very rapid, that the natural system has this property of being very, very rapidly degrading the food as if to feed the entire community. So the rate of degradation of particles by bacteria is really down to the rates of their enzymes. So here I have the question, how are the enzyme rates refined in these systems where you have these organisms that are producing all these different goods? So in this syntrophic systems. So here I have a cartoon that basically illustrates this process. So if you have this type of cross-feeding interaction, and then you have suddenly one of these types that, let's say, this yellow producing good type here on the top left mutates. So there's another new mutant that produces much more of the yellow good than the original ancestor producer. So this is this overproducing mutant. So an evolutionary question is asking, under what conditions would this overproducer replace the ancestral producer? Therefore, leading to a case where you have an overproducer which gives way more good to its partner, bacteria. And as a result, if the bacterial partners are more healthy and more vigorous, they would just by consequence produce even a little bit more of the other good complementary to the first one. So it leads to an overall more robust system. So again, because we have to frame things in terms of structure in order to understand any type of cooperation, then we're asking what is the type of structure that can lead to this replacement. And what's interesting about syntrophy in terms of degradation rate is that syntrophy, which is this interaction between these two goods producers, already implies that enzymes have to be faster if the systems are stable. And here is an explanation why. So in the case of a self-sufficient organism right here that produces all of the goods for themselves, their individual benefit maxes out at some point. So this relationship between enzyme rate and growth is a very famous relationship that's really well elaborated and expounded on in this body of theory called metabolic control that you may have heard of. But basically, it just says that you have increasing growth rate if you produce more and more of some kind of nutrient. But then at some point, the nutrient is no longer a limiting nutrient for your growth. So then the enzyme rate doesn't need to get any faster than this. So this relationship is kind of very easy to explain. It's very obvious. However, if you are producing a nutrient that has to then leave your cell and diffuse out and reach a partner, and that the rate at which it reaches the partner determines the rate at which you receive another nutrient, then that whole process slows down the equivalent amount of nutrient that you get relative to a self-sufficient system. So that means that prototrophs, because they have to overcome all these extra barriers of diffusions in and out of cell membranes, they have to have their enzyme rates much faster in order to achieve the same growth rate. And so if you have a system that depends on syntrophy to degrade all of the nutrients, that depends on different bacteria doing different types of degradation and releasing different types of nutrients, then just switching to this system alone from a system of self-sufficient organisms leads to much more rapid degradation. So that's where that's where the crux of our explanation lies. So let me summarize a little bit of our approach before then I move on to Ali, and Ali will present the next part of the presentation. So the rate of degrading, so here I have a cartoon of a piece of debris in the ocean. So that's a marine snow particle. And our bacteria that are attaching to the particle, and then they start to cause this degradation of the particle, and this gray cloud is this plume of extra nutrient that's produced. So this schematic is exactly as I've already described. And this schematic also illustrates a bunch of rates that we're interested in that maybe will help us to think about what controls evolutionarily the tuning of this degradation rate V, the degradation rate of the particle. And maybe help us explain how come this rate seems to be so high as to not be related to the growth demand of the attached bacteria, this attached population. So this is what's called in the literature, actually it's even being called the particle decomposition paradox. And this name was given to it by a bunch of climate science researchers who were surprised to discover that the particles are degraded so fast because it meant that they had to rework all of their carbon cycle models when they were trying to understand how carbon is moved around in the world, in the ocean system. So we want to explain this particle decomposition paradox as a cooperative phenomenon. And we have various types of evidence that we consider in the course of our exploring this topic. For example, we look at the distribution of enzyme rates, and there's quite a good body of work to support that indeed aquatic enzymes are aquatic degradative enzymes. So these extracellular enzymes of the type of enzymes that you would need in order to liberate nutrients from ocean particles are indeed enriched and highly active in aquatic environment. So we were intrigued to find this, and also this whole process of, because it's so important to climate scientists, there seemed to have been a lot of funding and a lot of support that led to scientists finding clever ways to determine all of these rates. And that seems like a very crazy feat, to be able to determine the rate of attachment to the particle, the rate of detachment. So I didn't provide a full explanation here, but maybe I could just elaborate a little bit more. So the bacteria that are swimming in the ocean attach to a debris particle, a piece of particle, and then they start to degrade the particle, and then they can release their progeny or dissociate, detach from the particle and live in this cloud of nutrient, which is called the plume. So there's a population in the plume. And then there are, additionally to that, there are bacteria that will just go straight for the plume. They will not bother attaching. So these are these are the more selfish type of bacteria that will just chase the particle as it's sinking down the water column. So there are scientists who have actually found ways to estimate various rates from this schematic diagram. And I found this really surprising. And it ended up being an effort where I tried to summarize and estimate these parameters from so far I have about 25 papers, but this list is still growing. And we intend, part of our approach is to simulate this entire process using parameters that have been estimated from the literature. And I'm still surprised that I could find and estimate all of these parameters from the literature. So it's been a very interesting process, and that's part of our approach. And then another approach is to try to look at particle degradation in a very simple conceptual way. And here I'm just gonna give it over to Ali. So I'm gonna stop sharing, and Ali, you can.
Wait, can I add something to just what you've said here, just to kind of as a transition, is to say that kind of what Dan noticed was, there's this question of how do you get these codependent, cooperative interactions between species and between individuals coming about. And in kind of the ocean environment, the question here was, what Dan noticed was, well, we need some sort of structure. Are these little pockets of structure sufficient? So that's the kind of question we're asking is, is this structure enough to explain the kind of dynamics that we're seeing? So that's what we're trying to figure out, is here's a well-mixed environment with little pockets of structure, and does that sufficiently explain the dynamics of between these bacteria?
Yeah. Thanks, Ali. Yeah.
So yeah, so I should say that me and Dan have been through many iterations of this model, and we are definitely in the process and like actively updating the thinking. So a lot of my thoughts are not finalized, and a lot of like the thinking is we're just trying to figure it out. So yeah, this is just a lot of like think of this as reflecting on the process of coming up with such a model is what I'll be talking about right now. So one thing that Dan has been talking about is us trying to figure out how do we start narrative descriptions between this idea of cooperator versus defector versus, you know, producers versus non-producer. So when should we use cooperator versus defector, and when should we use producer, non-producer? And personally, I came to the conclusion it's not that big of a deal in terms of the narrative consequences, but I personally prefer to use the concept of cooperator and defector when the strategies can change with the environment. So if the individual strategies change with the environment, then in kind of my narrative sense, it makes more sense to use this term cooperator and defector. And that's basically because, you know, you say that the individual is reacting to the environment and deciding what to do. And with kind of thinking about how should you frame the level of selection, individual versus group. And again, there I found that it's not too important. It's good to jump between the two. But personally, I found that I most like it when the selection is on the level that the characteristic is understood. And by that, basically, it's, for example, to explain flocking behavior, it's difficult to explain flocking behavior as the sum of individuals because what you're observing is the flock. Or a more easy example is to understand a warning call, you need the idea of the group. To just understand, to describe it to begin with, you have to start with this idea of a group and to say warning call. To try to describe a warning call on the basis of individual, which you can, it'll just be a lot less intuitive in terms of what we observe about the world. So that's kind of the intuition of saying these are the narratives that I personally prefer, but I don't think it's necessarily too important. So with the model that we're working on, it's basically, as Dan has said, is when you have this evenly shared public good, the free riders will always take over the population without any structure. So if you have zero structure and the public good is always just shared amongst everybody, free riders take over, or people that are not producing it. So here's an example of kind of wrapping our head around how can we think about this. And here we have a population where the green are producer and the blue are non-producers, and the light green shows the immediate diffusion of food produced by the producers. So if the food is immediately distributed everywhere, then the non-producers will of course take over. But let's say that there's some sort of diffusion rate and this blue out here doesn't get any of the food. Therefore, this means that this blue is growing slower than these greens. But so also we're imagining that the green has to pay a cost, some growth cost for creating this food. And the blue doesn't have to pay that cost, so it grows faster when it has food access. So basically, we find that on an individual description, you say that, well, these greens are growing slower than all of these blues, except for this one blue outside. So these greens are growing faster than this blue, but they're growing much slower than the blues inside the green. So as the spatial structure gets more defined, we see that, okay, now there's more greens that are growing faster than more blues. And you would imagine that in this circumstance, the greens will grow. So this is kind of a way to start reflecting on how spatial structure creates this differential between individuals within a group and individuals within another group. So another way that this is described and it's approximated is with Simpson's paradox. And Simpson's paradox basically describes the circumstance where you have this large population. The large population is broken down into some groups, and those groups have different amount of producers. And because they have different amount of producers, those groups grow different amounts. But notice how in each group, the non-producer always expands. But despite the fact that in each group, the non-producer expands, when they all come together, the producer increases in frequency, right? So this is why it's a paradox, is because in this intergroup time, in every single time, the non-producer is always expanding, but when you separate them into these subpopulations and you allow them to grow separately and then bring them back together, the producers increase in frequency. And this is, at first you might say that this isn't, this is similar to breaking the population up into 3 parts. So you are kind of introducing the structure for a second, and then you're taking away structure, is a nice way to think about it. But notice that if we spend too much time in this intergroup period, then the non-producers take over regardless. So there has to be this there has to be the right amount of time spent in this middle phase to allow for the producers to expand. And this also depends on the functions, but in general, if you spend too much time in this intergroup phase, the non-producers take over. So here's a kind of example to help our thinking. This is just a simple circumstance where you have a population and it breaks down into 3 groups. And in those 3 groups, you just figure out how those 3 groups contribute to the population growth. So here we have an entire population. It can either break down, if they can either be grouped into pure cooperative groups, half cooperative groups, or completely non-cooperative groups. And the frequency which that happens is just proportional to the random probability of them meeting. And here we have the payoff matrix, where it's if a cooperator meets another cooperator, they get 2 benefits subtracted by the cost of being a cooperator. And if a cooperator meets a non-cooperator or non-producer, then you get the benefit subtracted by the cost that you're a cooperator. And if a non-producer meets a producer or non-cooperator, notice how I'm having a hard time switching between over. Then you just get the benefit, which is better. And just looking at this, we can set up the replicator equations and get some simple dynamics about how this population will behave. And this basically shows us that when the population is below half, then the population, the defectors will take over. If the population is above half, and if B is greater than C, then cooperators will take over. And this is a very simple way to think about this, is because if B is greater than C and if P is greater than half, then if you're a cooperator, you're better off meeting another. Like if you know your opponent is cooperating, you're better off cooperating than cheating. So it's no longer the prisoner's dilemma. Therefore, you can defeat that system. But here we have the prisoner's dilemma, because if your opponent, if the person you're matched with is cooperating, you're better off cheating. So you're going to go extinct. So now we imagine the circumstance where we have 2 populations or a bunch of populations that are in this circumstance. So you imagine this scenario, but two of these populations that are going towards zero. And depending on what their initial frequency is, so imagine these kinds of populations, depending on what their initial frequencies are, they're going to go to extinction at different rates. And when we bring them together, we might see something like the cooperators increasing or the cooperators decreasing, depending on how long that intergroup time is. So with this model, a way of, sorry, I'm trying to make sure. A way that we've approached it is by saying that this is a very, very simple version of this, where we say the particle arrives and a certain number of initial initial producers and non-producers surround the droplet, and then they just start growing at an exponential rate. And if you create this model, you get something like this, where you have the resource, which is the droplet, and the food being produced by the producers and then being eaten by the producers and the non-producers. And here you see the food is being produced by the producers, and as the food increases, but it's shared evenly, the non-producers increase dramatically and then hit the maximum amount, which is the amount that you can grow given the amount of resources that you have. And that would be the end of the droplet. So this is kind of where we're at right now in terms of trying to figure out how can we use simpler ways of thinking to approximate the complex dynamics here. So the next step for this would just be to do a binomial distribution of all of the different starting frequencies of producers at each droplets, given the frequency of the population, and then sum those, and then find the frequency at the population level again. Basically do the Simpson's paradox example. And notice that depending on when you end the time here, you're going to get different results. So if we stop it here, you notice that this is going to be better for the cooperator because it's closer to its maximum than the non-producer is. So the non-producer grows more relative to itself past a certain point than the producer is growing relative to itself. So there is an advantage to having the time be stopped at the right moment. So going forward, what I guess we wanted to discuss with you all is, you know, one things that we need to include in the model to really capture it is arrival order. So right now we don't have the fact that maybe you have producers surround the particle earlier than non-producers. So you don't have a binomial distribution. You have a distribution that is a little bit more favoring of producers. And then and also what's, you know, in this plume area, are the producers and non-producers exchanging with the environment and at what rate? And really the difficult part for me is trying to figure out, how do I mix this game theoretical concepts that are very simple with the analytical portions here, and what is the balance between a analytical description that is simple enough to actually get decent results, and then use that to ground the simulations that we're going to do. So that's kind of an introduction to our modeling approach. And yeah, Dan, do you have anything to add?
I just have some slides about future directions, but I think that maybe first we should see if there are any questions.
Yeah, if there are questions so far, or everybody is willing to look at the future directions. I think you can complete the future directions, and then we will have something like 20 minutes for discussion. So okay. It's best that you maybe quickly go through that.
I'll just in one slide kind of give you a little bit of a taste. This is another aspect, I guess, of the future directions, because one of the future directions, as Ali mentioned, was to make the model more realistic in a dynamic sense over time. So having different arrival rates is a very important aspect of it, and modeling also that cloud of excess nutrients, the plume, and having organisms go to the plume is a very interesting addition that we're also considering. But here I thought that this, as people who study reaction networks and stuff, I thought that you might be interested about this, because as part of my research and summary and trying to understand how bacteria share nutrients, we kind of realized that there are different social interactions on a biochemical level. So previously, when I'm making these diagrams of goods, this is what you may call an evolutionary diagram, actually, not even a real biochemical diagram, because you're just showing benefits. But this is more a bit one extra layer of complexity or detail. And here I'm showing three different types that I think I'm separating into types because they are quite different qualitatively with regards to what we're talking about. This is not an accepted division, but I think this is a very logical division that we are proposing. And each one of them has natural examples, and I can elaborate further too. But here, let's just go one by one. So these are supposed to be two syntrophs, so they are exchanging molecules, metabolites, but they do so in different ways. So starting from one chemical here, this green X, if there is a scenario at which the nutrients, these stars, are just simply different nutrients for the green organism and the red organism. So the X is converted into a nutrient, and then there is some kind of byproduct, like a waste product. And the waste product in many cases actually inhibits the growth, because it's the waste product of the green organism. And so the growth of the green organism can be relieved by the presence of the red organism, which takes this byproduct and actually uses it as a source of energy or food for itself. And one very classic example is that anaerobic fermentation leads to hydrogen production, and hydrogen production is very inhibitive to fermenters. But if there are methanogenic archaea in the mix, these methanogenic archaea use hydrogen in order to produce methane. And so this has been, they've become so dependent on each other that all fermentative anaerobic fermenters produce these copious amount of hydrogen that they cannot deal with on their own. So they almost kind of assume in their physiology that there's going to be a methanogen around to help. And then just moving on to the next one, this is a little bit more similar to the diagram that I showed in the beginning, where you just have, let's say, two different polysaccharides, two different complex chemicals like this red dots and this green crosses. And each one gets turned into a different nutrient. And then these nutrients are shared between the red and the green organism. And this will be like an exchange. And then finally, it could be in some scenarios, this is a very interesting and perhaps understudied and also interestingly vulnerable, but has many interesting properties, this type of kind of a production line or a sequential cooperative pathway, where the green organism actually doesn't produce a nutrient directly, but it produces the intermediate molecule that then gets converted into the nutrient by the second organism, and then this nutrient has to be shared by both organisms. And the interesting thing is that these are all examples of syntrophy or cooperation in terms of sharing metabolites, but they have completely different consequences if they're supported by structure. And also they have different requirements for structure. So on the left here is this partitioned example. You don't actually need structure at all, because it's and the good example that I can provide is an analogy in animals. This is kind of what's called a classic huddling for warmth type of cooperation. And what does this mean? It means, at least if you're an animal, you cannot cheat by not producing warmth as a warm-blooded animal. So if you're huddling together for warmth, you don't suddenly turn off your warmth. So there's no really way to cheat in this social interaction of huddling. There is individual benefit for every individual that comes together to huddle. So in this way, it's very much the same way. There is individual benefits for doing the individual tasks here of the cooperative degradation for both the green and the red organism. And they can become dependent on each other, and they're cooperative with one another, but they don't have the cheer problem, because there is no way not to use the green in order to eat. There's no way not to use the red in order to eat. And there's no way not to help the green by relieving him of his waste product for the red. So in this way, it's kind of this partition metabolism is sort of highlighting that there is some kind of biochemical difference between these two organisms, and that also protects them in a certain way. But with these two cases, as would be the case of degrading waste on a marine snow or a particle of ocean debris, they're all the different polysaccharides. They're all different from one another, but as I mentioned, you can also be a self-sufficient organism that does everything. Here, maybe it's just important to note, you cannot do everything, because this is your waste. This is kind of you're producing something that's antagonistic to your type, your biochemistry. Whereas here, these kind of things, because you can do both steps, because they are so biochemically similar to one another, you can kind of switch between one and the other. You can lose the abilities. You can just receive the nutrients but not give back. And so there's a fundamental difference between the one on the left and these two on the right. And then I'm further separating them because the sequential one is just turned out to be very interesting. We can talk about it more if you would like, but it just requires a lot more support, because this green organism has to do this weighting, whereas here it's kind of more symmetric relationship. And so both of these, just to make a long story short, do require structure. And when there is a structure to support them, they end up producing more, and so leading to faster degradation rates, because they do benefit from the structure. Whereas here, you don't necessarily benefit from overproduction. It actually can be dangerous. And so on that note, we can close the first phase of the talk, I guess. Yeah. And thank you.
Yeah, thank you, Dan, Ali. Yeah. Really interesting topic. And to me, I will open the question round, but I just want you to give your impression on this this word that also Ali used a lot, the word narrative, which is like what you say in the beginning. How come we think that microbes are little humans making decisions? We know they are not. So have you ever come up with some sort of ontology to speak about them, what they mean, or what their intentions are, or something like that? Like, or do you think these notions are just like completely unnecessary, and we attribute these things to them? Or like, just wanted to talk about want you to give a general view on the notion of narrative about this, and the social. Are microbes social beings because they eat, because they share goods, and so
right.
Yeah.
Oh, there's a lot there, Thomas.
Yes, just a glimpse, if you can. If it's too much, we can leave it for another session. Yeah.
Yeah, it reminds me. I just saw a talk recently by a very famous social biologist of not bacteria but amoeba, and she was showing a small video of an amoeba eating a bacteria. An amoeba is a large blob, and a bacteria is a small blob. And then you see the video, the amoeba just goes past and then eats the bacteria. And then she's telling them, guys, just imagine it's the cheetah and a gazelle. It's just a cheetah eating a gazelle. So I think she was trying to not just trying to make it more interesting for people and more relatable, but I think relatability and interest is kind of like just the first tools of engagement conceptually with things that we just are so far away from us, like bacteria. And I think it's been really telling for me that the most satisfying solution that I found for bacteria. So organisms, much of sociality has been focused in terms of what the organism can do. So sociality is usually solved in bacteria or viewed in bacteria in terms of are you appearing to be like an ancestor of a multicellular organism? Are you and your daughters and your clone mates interacting in such a way to create this group that's very cohesive and so starts to act like a multicellular organism? So that's how they think of sociality in microbes. And so it's always what the active role that the organism plays. So you don't even have to think about the physical environment in such a way. And then in animals and in plants and in all the higher organisms, it's really even further removed really from the physical environment. There is something, I mean, it's always there, you can't really ignore it, but there's a much more active role of behavior and behavioral choices, and that's where the whole word strategy comes in. But I think what's been really telling for me and really about why this whole way of looking at it is so satisfying is that here we're just saying that the bacteria are these thermodynamic particles that are just being controlled from the top down by the environment, and that it's the environment that provides, that nurtures and scaffolds and provides the basis to create what appears to be sharing and cooperation in the bacteria. So that's sort of not, it's the main difference is that we are not looking at something active that the bacteria are doing, but we're looking at the way at which the bacteria are arranged by the environment. And I think that that's very interesting and satisfying for me to look at it that way.
Yeah. Thank you. As you said, this opens up so many things. But I would like to give the word to others in the audience if you happen to have a question. And this last slide is very interesting. We shall discuss like at some point, because these look like just different reaction networks to me.
Right. I thought so.
So anybody has a question? I think someone unmute in himself.
I did. I was just gonna add that with this discussion of, because this is definitely something that is very, was a thing that I had to combat, of trying to figure out how to think about these narratives. And the solution for me was just when I thought, well, you always start with what is intuitive as the description, and then you acknowledge that you're making something into a metaphor. So like a wolf pack hunting is clearly cooperation, because that's what we mean by the word cooperation. And then as you go away from the most intuitive definition of the word, it becomes a metaphor. So I think it's one of these things that you have to acknowledge that it's similar to the group selection versus individual selection. It's weird to describe a warning call outside of the context of a group. The same way it's weird to call something cooperative, but it's useful. So it's useful to think about a warning call from the perspective of an individual at times, to flip the perspective away from the most intuitive way of describing it. But with the cooperation thing, I think that it really is just a metaphor. Like you're developing a tool of consequence. You're saying, if I can organize the world in this way, there are known results. Therefore, let me see if I can describe it in a way that has these known results, while acknowledging that it's only simply a metaphor when you're taking it outside the context where it was intuitively described. So I think it's a lot less of a problem. That, at least for me at the beginning, I thought it would cause a lot of problems. But I realized that there's more of a comfort of recognizing where you're in line with the intuition of observation, and when you're using metaphors to reorganize information. So you're taking your observations and you're placing them with kind of known associations in other contexts. And you're saying, okay, well, do associations that I've seen elsewhere apply to my context, knowing that those associations are not what you're seeing right now. Like, you're not looking at the bacteria and you're thinking cooperation, but you're thinking, well, maybe if I did think about it that way, I'll find something. So I think it's just a nice tool to have to apply that way of thinking onto something that doesn't immediately, it doesn't immediately tell you to look at me through the lens of cooperation and defection. But if you do, you might, for example, a very simple example is that thinking about the prisoner's dilemma is very easy. So you just think, well, if you are in this hypothetical state of a well-mixed environment, then you're going to be in a prisoner's dilemma. If you're constantly in a prisoner's dilemma, then you're going to go extinct. So it allows for a kind of simple way of thinking, given a genre of investigation that has been taken to its natural end. So I see it more as like a tool and moving away from intuition and then using new tools. I don't know if that helps, but it helped me be more comfortable.
No, definitely. It's good to make this distinction. Okay, anybody? Diederik?
Very interesting. It makes me also think, but I've not read myself about it. I should have a look to this article. But you remember, Thomas, you even organized that talk of Kranikov, where he referred to an article where they kind of looked to decision-making of bacteria. I think Kranikov worked a little bit on that. I don't know if it's serious. And that, of course, would be more like real decision-making on the level of the entity, which is the bacteria. If I remember well, they saw that there was a conjunction fallacy made by the bacteria or something like that. But I've not read myself about it. I only saw a reference to it.
Yeah, me too. I only recall the reference.
You remember it was in the talk that you organized with Kranikov, I think even.
Yeah, he mentioned it. The talk was
he
more.
Mentioned it.
Yeah. He just.
Yeah, he only mentioned it. But it would perhaps be, because it is true, of course, that these paradox-like situations, that's exactly where one could suspect that something else is going on. One should not be afraid, I think, of this possibility. It depends a little bit where you work, of course, also, whether you could think of bacteria who make kind of primitive decisions themselves or not. The example of the spring, I think you could look at it as if you connect the two springs, you kind of destroy some possibility for interference, for interference of the two springs, I suppose. That's perhaps a way to look at it, which could be a hint to the fact that there might be a negative interference play a role, a negative interference between entities, even if they are bacteria. They are very similar to decision making, like it is studied in humans. Huh?
It's interesting.
Even human decision making is not. It's also metaphorical in some way. Yeah, always.
Right. Mm-hmm. When you mentioned that, it makes me think of there is something interesting that I found while researching this process here, because one of the things that we look for when we construct this model is that those organisms that are degrading the debris are paying a cost, right? And the cost is pops up in the game theory as well. So naturally, when you have this natural example, then we are okay. So this bacteria is the one that produces the enzyme. What's the cost it pays? So can we look at the growth rates and see that the bacteria that produces the enzyme is a slower growing bacteria, and then look at this difference, and that will be our cost. So that's kind of the intuitive, or how do you say, the would be the most straightforward way to try and go about looking at this bacteria. So I went ahead and I tried to Google and do some research about these different types of bacteria and what's the cost of producing the enzyme. And then I found that from the little evidence that we have, actually it appears like bacteria that produce the extracellular enzyme have this ability to have a physiological switch. In other words, that they, as soon as they are starving, they look for a particle to attach, then they attach, then they release enzyme. And at the moment that they release enzyme, they do pay a cost, they're slower growing. But as soon as there is an excess of food, they stop producing enzyme, they detach, they go to this cloud, and then they appear to grow just like the so-called cheater. So they can switch between an actively producing sacrificial pro bono degrader and a fast consumer that looks like the cheating kind of type. So they have this ability to have this physiological switch, which I thought is very interesting. But if you wanted to, I guess if you wanted to consider that as a sort of a decision making, you would have to juxtapose that to another bacteria that may, I don't know if that's kind of how you were thinking, because in bacteria, at least, if you are able to make this physiological switch, you always have to compare it to bacteria that can't make this physiological switch. So it's almost like having a decision-making ability versus not having a decision-making ability.
Yeah, definitely. But once you introduce this possibility that they can make a choice, it probably will not be deterministic. This means that on the statistical level, you will have all kinds of effects that often are paradoxical. Huh? So it would be interesting at least to look at it. Most of the paradoxes in human decision-making, they get clarified if you really allow decision-making to have a certain structure. I don't know if you're a little bit knowledgeable about all these weird paradoxes that appear in human decision-making. The conjunction fallacy, they're called often fallacies, yeah? But once you understand the mechanism of decision-making, that it's not a Boolean mechanism, for example, that it's not a logical mechanism, but it's much more a mechanism that has to do with recognizing similarities and much more complex than just Boolean logic decision making. Yeah? Bacteria are certainly not mathematicians. That I think would be a bad hypothesis. But they might have tendencies like people have when the stock market is raising, and that there is a bank run, and these kind of decision making that are not logical, that are more linked to if you look to the mathematics as at play, you have more vector space structures and similarities, which is also the reason that, for example, computer scientists use that in artificial intelligence. Every search engine uses actually similarity structures in vector spaces to look to things that are close by, and that kind of structure could well exist on the level of bacteria, I think, which is a kind of a more fuzzy-like decision making.
Yeah, it's interesting. I mean, just I was wondering if someone else has a question as well, maybe Francis or someone, because we don't have time for a very, very long discussion. But just to complement what you say, Diederik, it's very interesting to think of how the architecture of the bacteria can implement these functions. Let's say switching or computing as in the way that like a vector space similarity function. And to do this, we need to think of the let's say biochemical architecture of the bacteria as a computer. And this metaphor is useful perhaps to see if it's able to implement some sort of RAM memory or some in a different way. And there is an area called chemical computation, I think, which has done some steps ahead on this line. So this is another, only if you want to increase your slides in future work, then we can keep going. Yeah, but I wanted to ask if someone else has a question or a word, because we don't have much time and it will be good to have as much feedback as possible. Yeah. So Christian, Francis, I don't know.
Yes. I didn't see the beginning, so maybe my question has been answered, but I noticed that an interesting concept you use, one that I didn't know, is the one of syntrophy, which seems to be similar to mutualist symbiosis, but which has the idea that the one produces what the other one consumes and vice versa. So there is a cooperation between two types of agencies. But I think it becomes much easier to be cooperative if there are several types of agencies, one of which produces something that another one consumes, which then produces something else, so that it's not a one-to-one exchange, which implies a much more limited kind of cooperation, but it's more that a many-to-many cooperation, like some bacteria species produces something and consumes something else, and then others do the same. And as long as the whole forms what we call a chemical organization, that means that everything that is consumed is also produced, then the whole thing is symbiotic. So you don't need to have the one-to-one symbiosis or syntrophy or mutualism. So I don't know whether that was implicit in the model, because I didn't see the beginning of the explanation.
Right. Yeah. So it's a good point. Maybe part of the clarification. I don't know if this is kind of along the lines of what you're thinking, but at least there is yet another set of terms in which those, so as I mentioned in the beginning, so these particles of debris, they are aggregates of all sorts of types of polysaccharides. So there's some dead plankton, some fecal pellets, all sorts of different things, larvaceans and stuff like that. So there are all these polysaccharides. So and each one of them has slightly different nutrients once you liberate them. And so there's so many different enzymes that you need in order to degrade all of them together. So you end up having these organisms that can degrade a very large array of them, and some organisms that can degrade only a subset of them. So another way to think of these syntrophs is kind of like specialists versus generalists. So ones that are specializing in degrading a very particular type of polysaccharide. And so they can provide for themselves and for others a large amount of a certain type of nutrient, certain type of sugar, and they can receive amino acids from a specialist that degrades proteins very well, or polysaccharides that have protein substituents inside of them. So then you can kind of get something that's, I mean, the choice to think of it as a one-to-one is just isolating for simplicity, but it's really, as you say, it's a many-to-many.
Okay, thank you. I think Christian also wants to contribute, and definitely just as a, well, final thing, we might continue this discussion. We will discuss about it later. But yeah, Christian, please go ahead.
Yeah, Well, first of all, thank you very much for this presentation. I have two things. I just want to add something on the narrative topic. It's just because I'm teaching at a school for communication design, and so I find it super interesting that you make up your mind really about what could be the right narration, because this is for communication design. This is at the heart of the matter. What we try to do, finding the right lens, you could say, through which we look at things. So finding the right metaphorical, the right metaphor through which we can describe things, to which we can explain things. And it's always the problem, I would say, that looking through one lens opens one door of understanding an object and closes at the same time different other doors. And this is always so it's really about trying to think what I want to achieve with my choice of the metaphor, with my choice of the lens that I'm looking through. So this part I found really interesting on that level, and really inspiring. And the other thing is, what I find super interesting is your whole question of spatial structure and how spatial structure arranges the behavior of the bacteria, because what we're experimenting with in my seminars is, for now almost a year, is that Thomas introduced us to chemical organization theory, and we try to describe, this is the goal that we have. We are not there, not at all. We are still trying to understand the whole reaction network scheme, and how we can apply this to design processes, to describe design processes. And one of the interesting things, or one of our discussions we also have with Thomas a lot, was that with COT, the whole spatial structure is calculated out. And we found that for design decisions, we need the spatial structure. So in the last semester, we tried to model with a reaction network a visit in a supermarket, and then the decisions of what do I buy and what do I want. And so, and we did this because of the spatial structure of the supermarket. And how can we get this in? And what does a supermarket structure? How does it conduct our behavior, our decisions, and our thinking, and our feeling, and all this? And so, from this point of view, I found your presentation super interesting. And just for clarification, I have my last point, a question. Did I understood you right that you looking for structures that support the behavior on different levels, so that on the one hand you have spatial structure, and on the other hand you have also interaction structures, and then as a third point, sequential structures like the arrival order and things like that. Did I understood this right?
Yeah, the spatial structure will support different interaction structures.
Okay. So okay, but you don't look at them in as completely different levels, so that you can work out an interaction structure without thinking about any spatial structure, but that the spatial structure always.
It's an interesting point. I think it makes me, maybe this is kind of similar what you're saying. I was thinking about this when you're talking, because this is an indirect relationship to this, but something like if you have a network, the effect of spatial structure, in a way, I don't know if you can have a direct translation conceptually, but the effect of spatial structure is to add and remove certain abilities of certain edges, you know? That structure makes me more likely to interact with my type if it's a very limited diffusion environment. So it makes certain edges much more likely than others. So maybe it's something that you can incorporate into COT, but you have to understand how spatial structure, your idea of spatial structure, changes the network, sort of.
Yeah. Great, thank you.
And it matters what you define as structureless. That's the, I think, a big key feature.
Yeah.
Okay, guys. I think it's very good to have had this moment with Dan. To me, a long-standing waiting for a long time to manage to get this thing happening. And your research topic, Dan, I think it's relevant for all of us, systemic modeling, and also for CLEA in general. So maybe in the future, there will be a chance to invite you to give a more general seminar to the whole CLEA people, because I advertised it very, very kind of, not very much, I will say, only within the group. It's easier for me to do it this way, but maybe in the future we can invite you to give a bigger seminar. Maybe even to visit the group, which is also something that will be great to have you around for some time to discuss about this goal directedness and your take on this stuff. So yeah, with this, we say goodbye, and we will be meeting in 2 weeks again. I will include Dan and Ali in the list, so you can know what we are doing and participate if possible. And one small big thing, please share the articles you mentioned here, because I think they are very relevant, especially the one in the next slide, the one by De Souza. This one, I think it's very, very useful for us to understand this, the way in which bacteria spatially structure, because that is pretty much like the idea you had, like how space provides the means for certain things to emulate what we think of decision making in a certain way, strategies, or so it's interesting, this action and spatial structures merging together are probably the best architecture for something like, as we were saying, like decision making. So it's kind of like the Turing complete thing, the action network plus the structure. So it's interesting to go deeper on that. I'm sure that this guy wasn't thinking about that when writing the article, but for us, I think it's going to be maybe easier to get close to that perspective. So yeah, very interesting once again, and thank you very much. Clap, clap, clap. So I'll stop the recording.